Okay, so I'm very grateful to Ellen and Ute for inviting me back to Ben-Gurion for another episode of this conference. It's wonderful to be here and to catch up with some old friends and meet new ones. I never met Eric Davidson, but was much influenced by his writings and engaged in a very stimulating exchange of emails with him. I was very disappointed not to meet him, but Ellen's beautiful description of him brought him to life earlier today, so for that, thank you. I thought I would just begin by telling you about what I'm not going to talk about, just so that you know that I actually do have a day job. Partly out of a sort of nervousness about what I am going to say, because it's going to have, I'm not going to really discuss much of what we do, or have done, but what we would like to do and why we think it's important. I'm a little apprehensive about that, but I would really welcome your feedback about whether this is actually worth doing. Okay, so we think about cellular information processing. We've worked particularly on post-translational modification and on gene regulation, and on developing some of the sort of mathematical underpinnings that are necessary to work on that. But more recently, in the last, just before the pandemic actually, I've become very interested in this question of learning, and particularly at the cellular level. And much of what I'm going to talk about is taken from a survey paper that's just actually appeared in a place that no biologist will actually read it with probability 1, the proceedings of the IEEE. So I'll mention it here. It's open access, and you can actually get hold of it if you find it interesting. This is work that kind of emerged from a collaboration with a colleague at Harvard, Sam Gershman, who's a cognitive neuroscientist. We've written this perspective in life that came out a little while earlier. And the starting point for all this actually goes back a long way, at least in my mind. The sort of proximal spark for this is a piece of work that came to fruition after about 10 years in the lab, which I'm not going to talk about again. This prefaces all the things I'm not going to talk about. But just to say, so this is the Lieb Center that Ute has created here. And back in the day, Jacques Lieb had a famous debate with Herbert Spencer Jennings, basically about the nature of life. Jennings put forward, made some remarkable studies of unicellular protists, ciliates, and described a remarkable series of avoidance behaviors in one of these ciliates. These experiments were later deemed to be not reproducible by the experts. And the short version of this story is that the experts were wrong. The experiments are reproducible, and we reproduce them, and you can now observe the videos to see for yourself. The moral that I wanted to take away from this was that when I read the literature in the 60s and 70s about this question of the capabilities of individual cells, all I can say is that the consensus that single cells were incapable of the behaviors that Jennings ascribed to them was basically ideological, in the sense that people preferred to believe some really shoddy experiments because they said the right thing, to some really good experiments that said the wrong thing. And this was a warning for me that this area that we're going to enter is rife with ideological minefields. So consider that a trigger warning. We're going to try to stay away from them, but they're lurking all around us. All right, so I think we all have an intuitive sense of what learning means. So let's go with that for a slide before getting into it. I wanted to sort of step back a little bit and say why I think it's interesting to think about the question of learning in cells. This is sort of what I distilled out in terms of reasons. So the first thing is that learning is really a very universal principle, process, which cuts across biology in the sense of organisms and cuts across scales within biology. And there are not too many such principles in biology. And I think that brings learning particularly to our attention as something that should be interesting. And I think, particularly in the light of the theme of the current conference, one of the things that I find very attractive about it is that it allows us to avoid falling into a very common, what I think of as a pitfall, which is thinking of constancy and plasticity, to use Ute's words, as antinomies, as being fundamentally opposed to each other. And I think another opposition that we're all familiar with is the one between nature and nurture. And the point about learning is that these two things are inextricably part of learning, because there is the mechanism of learning, which is essentially constant. It comes to us from nature, it is often inherited. And there is the content of what is learned, which is, of course, a reflection of how the organism is interacting with its environment. It depends very much on what's out there at the time. It's very plastic. It's always subject to change. It is about nurture, not nature. So learning allows us to steer a rather different path in thinking about these things that we often think of as in opposition to each other. So when I started thinking about this and talking about it in the lab, my lab members got a bit alarmed. I think they were asking themselves, why is Jeremy going off into the wild blue yonder here? And in my own mind, I actually see this as a very natural extension of our concern for cellular information processing. But I think the thing that makes it different is that it's adopting a fundamentally functional perspective rather than a mechanistic one. And to those who are familiar with Nico Tinbergen's sort of breakup of how we should think about biology, to me this forms a very natural part of that way of thinking, that together with the mechanistic, evolutionary and developmental perspectives, we need also the functional perspective, and learning gives us that. And finally, and in many ways I think for me philosophically the most important point here, is that it offers us a different kind of metaphor for thinking about living systems. Because I think the metaphor that we have felt most comfortable with in modern biology has been the metaphor of the machine. And we have a lot of machine-like words that we use to describe what goes on inside cells. We have clocks, we have oscillators, we have gene regulatory networks. We have computers, which is also used as an analogy. And that's all very well, but there's always seemed to me to be something slightly lacking in this in terms of life. And I think what learning offers is a way to maintain the rigor of the machine perspective, but to now imbue it with something new, which is this idea of a machine that constructs an external representation of its external world and uses that to guide its behavior. And to me, that gives it a sense of agency and purpose, which for the most part has been lacking in previous mechanistic views. So I think there is in principle a lot at stake here if one can pull some of this off. So very high level reasons for why. Ellen.
It's not a trigger warning. You're among friends. You just described a T cell. All T cells do exactly what you're describing, so carry on and know that you're being accepted.
Ellen, thank you. The point is I'm not usually among friends, so I appreciate that. Okay, so having sort of made all this pitch about learning, what is learning? So learning goes, so scientific studies of learning go back to the early part of the 20th century, and particularly to behaviourist psychology. Some of this will be very familiar to you. This is just a starting point. Learning is a very sort of open and complicated subject, but it's a very good starting point. We can still use it for purposes, for modern purposes. The behaviorist psychologists made distinctions between learning that involves basically a single stimulus, so-called non-associative learning, and the classic forms of that are a repeated stimulus gives a decreasing response, which we call habituation, and its analog, a sensitized response. And we'll come back to habituation later. These are often thought of as somewhat separate, but really, I think that they're often seen together. And the more interesting associative learning of two or more stimuli, and I think everyone here will be familiar with Pavlov's salivating dogs, and I don't need to unwrap that any further. And you might also be familiar with Skinner's work on what is sometimes called reinforcement learning or instrumental conditioning, and what parents have learned from a long time ago about how to sort of teach their children. You punish them when they do the wrong thing and you reward them when they do the right thing. It's not rocket science. However, if you haven't seen Skinner's pigeons playing ping pong, I strongly recommend looking up the YouTube video because it's actually really fun. Okay, so this is the sort of behaviorist primary initial sort of breakdown of forms of learning. And this idea that learning forms, particularly in the context of associative learning, through making associations in time between stimuli, fits very nicely with what the neuroscientists found was going on inside brains. And this is the idea of long-term potentiation that takes place between neuronal cells. If you have a presynaptic neuron which fires a pulse just before a postsynaptic neuron, then the intervening synapse becomes strengthened. And that's long-term potentiation. It's, the full story is a little bit more complicated, and I'll leave you to read the slides. But there is, again, this idea that a close association in time is the thing that's being learned, and here it's actually a change in the structure of the underlying molecular biology of the synapse. So this association between stimuli and between what goes on inside the brain was a match made in heaven, and it is still very much something that neuroscientists believe. When you ask them what is the basis of sort of learning and memory, it's changes to synapses. So part of the cognitive revolution was actually the realization that learning is not about association in time. And I'm condensing quite a lot of material here onto this slide, but basically the generation of psychologists that followed the behaviorists were able to show that learning, that association in time is neither necessary nor sufficient for learning to take place. And I think this definition that I've sort of put in a box here is an attempt to sort of extricate from quite messy and diverse literature a sort of definition that we could sort of at least live with for the purposes of this talk. What does it mean for a system to learn? There are two components to this. One is that the mutual information between the internal states of the system and the external states of its environment must increase over time. And that's one way of saying that there's a transfer of information from the outside to the inside. And when I say mutual information, that's something we can actually measure. It's a term that comes to us from Shannon's information theory. There's a second component to it, which is the internal representation of that transferred information has to persistently influence behavior. And by persistent, I mean over a timescale that's longer than the presence of the information in the environment. And I think both of these things are part of what we could say is learning. Now, we could have a discussion of whether this is adequate. I think it forms a part of some of these new cognitive definitions, but this, I think, is what we're going to use for the purposes of this talk. Okay, I'm going to skip that because in the interest of time. How much time do we have? I've probably started.
16 minutes.
16 minutes?
Okay. If you want to have a discussion.
Yeah, the previous slide was really, we can come back to if you're interested in, but had to do with why you need both of these statements to capture what we mean by learning. So one of the reasons I think this issue of learning at the level of the cell is interesting is because of one of the, I think, most interesting problems that current neuroscience is grappling with, which is the nature of the engram. What is the physical nature of cognitive memory? We all know, we have an experience of remembering facts, episodes, over a lifetime. So somewhere in the mush that we call our brain, there's a representation of that information. And when you stop to think about it, that's remarkable. And we have really no clear idea of what that representation consists of. Now, one of the directions that neuroscience has taken, it's not the only one, but one of them, is that associated to the changes that take place at synapses during learning and memory are changes in gene regulatory programs within the nucleus. And these gene regulatory programs are often long-term, in the sense of months in the context of experiments done with mice. And they are based on very much the same kinds of molecular processes, epigenetic molecular mechanisms, polycomb, and all these other things that we are familiar with from a developmental context. Now, in the developmental context, we have another form of memory. We sometimes don't call it memory, but it's a memory of self-fate. Right? Cells learn that they are going to become a particular kind of cell, and they remember that, in many cases, over the lifetime of the organism. So here we have two forms of memory, cognitive memory and developmental memory. And both forms of memory are extremely long-term. And both forms of memory actually use the same molecular processes under the hood. And I don't think that's an accident. Or to put it another way, I don't think we will solve the problem of the cognitive engram without solving the problem of the cellular engram as well. And so this is another reason why I think this direction is an important one to take. Okay. Very quickly, some examples of learning that follow the definition that I laid out. Some of these are obvious, and I'm sure you already have thought of them. Evolution can certainly be considered as a form of learning, and has been. And that statement is not just an analogy. One can make a formal mathematical relationship between models of evolutionary processes and models of learning. And Jos Satmari and Richard Watson have written a very nice survey on exactly that. It's a form of learning that takes place in populations, not individuals at the time. The other form of population-based learning, which we're very familiar with, is the immune system in vertebrates, for instance. And this is, we know it follows exactly the two requirements that I laid out. We acquire information from the environment about pathogens. We represent that inside the body in terms of the states of T cells and B cells. And we remember that over a lifetime in the case of some of those cells. And we understand a great deal about the mechanisms that are responsible for that. Here too, it's a population form of learning because it's thought to rely on clonal selection. The one I like most is CRISPR, which we know about, of course, in terms of its technological capabilities, but actually it's a beautiful adaptive immune system that occurs in individual bacteria. And here we see the whole process laid out for us because we know exactly what the engram is. We know exactly what the internal representation is. It's DNA. It's cut out of an invading phage. It's placed in the CRISPR locus, and then it's used to guide subsequent behavior. So it could not be a better illustration of what I set out. It's not thought of as learning, and indeed it seems that the word learning is sometimes used, but it's not really sort of used in an intellectual sense to guide some of the study that goes on in the field, but perhaps it could be. And now there are some forms of learning which perhaps are not quite so clear-cut and so obvious as learning, but I think are worthy of attention. We don't think of learning as taking place when we talk about adaptation and homeostasis, but when you look under the hood, and from a homeostatic point of view, a lot of under the hood comes from control theory. What we see there is that in order to control a system, you actually have to learn about the environment that is impinging upon it and construct an internal model to guide your control. So there's quite a lot of evidence for the existence of these kinds of internal models coming from physiology. I think the interesting one from the point of view of this conference is the question of development, and the question of how and to what extent learning takes place during development. So I think we can ask ourselves, from an information theoretic point of view, what goes on to produce the individual, and I emphasize the word individual rather than the organism, because I think it's reasonably clear that the information for the organism comes from the genome. But where does the information for the individual come from? Well, part of it certainly comes from the genome, because we have mutations that influence our individual characters. And we have a pretty good idea of how genomic information is organized, and Eric Davidson was one of the pioneers in understanding how that information is represented in the form of regulatory networks and so forth, which lead to the dynamical systems that James was telling us about earlier. We have a very good sort of mathematical and conceptual handle on that part of the story. But when we come to asking how the information internal to the embryo, which means the information provided by other cells as to things that we wrap up in morphogen gradients and all these other things, how that information is, what kind of formalizations are needed to understand that information, that's less clear-cut. And I think that's where the learning may be taking place in terms of what individual cells are doing in response to what they see from their neighbors. There's a component that everyone lumps into the heading noise, which means everything is molecular and stochastic, and therefore things just happen randomly. And then there's that fascinating other bit, which is all the information that's external to the embryo. And we have extremely good evidence that comes from epidemiological and clinical studies that these environments determine what goes on inside the organism. Adrian mentioned in his talk the critical periods during which those processes are known to be particularly influential in early development, but I think we have very little clue about how that information is actually encoded in the system. So I think this issue of how learning takes place during development, to me, is one of the very interesting questions to which I don't have an answer, but which I do think there is an answer that is interesting there. And finally, I think we tend to have another dichotomy that we talk about a lot between mechanisms that are instructive and mechanisms that are selective. And to my mind, this is just a question about how many bits of information are actually being used from the environment. And a selective process uses very few, and an instructive process uses many. And I think by taking this information theoretic view, we can see this as much more of a gradient rather than a dichotomy. Okay, that's all very well, but how do we actually study learning? And when Sam and I, my colleague Sam Gersh and I got started on this, we were very interested in the possibility that conditioning, Pavlovian conditioning, was actually possible, took place in single cells. And I still believe that's true, but it turns out to be quite difficult to approach that experimentally. And what we realized is that, in fact, we really don't understand an even simpler form of learning, the most simple form there is, which is habituation. And so we have kind of stepped back to think about this afresh. And I'll just run through a little bit of why this, I think, is already a very interesting and challenging problem in its own right. You might think habituation, single stimulus. I mean, how hard can that be, right? Okay, so back in 2009, a very eminent collection of neuroscientists, ethologists, psychologists got together and put together basically a consensus statement about what they believed habituation is. And it consisted of these 10 characteristics that habituation exhibits in a basically extremely large range of organisms. We'll touch on some of them in the upcoming slides. But this is not just vertebrates, it's invertebrates, and all sorts of different behavioral paradigms in which habituation takes place. I won't run through them other than to point out that some of the more interesting core features of habituation are what happens when your repetitive stimulation increases in frequency. Well, what happens typically is that the speed of habituation increases. Rather paradoxically, what happens if you decrease the intensity of the stimulus? Habituation also happens faster. So these frequency and intensity dependencies, again, are observed over a very wide spectrum of organisms and paradigms, and are part of these characteristic features that are observed here. Okay, so I said that habituation occurs very broadly across organisms. This is just an example. They occur in Kandel's famous experiments on aplysia. This was the starting point of uncovering the mechanisms of learning in neuronal cells. There are studies in single-celled organisms, in particular in ciliates, very good studies that go back to the 1970s, in which, again, the characteristic features of frequency dependence and intensity dependence were shown in this single-celled organism. And then, to my surprise, there's a whole literature in neuroscience under the heading of repetition suppression. Perhaps there are neuroscientists in the audience here. What is really peculiar about this literature, because it's very much about exactly what we're talking about, repeated stimulation giving rise to a decreasing response. This is observed at many, many different levels of the brain. It's observed in the periphery, in the sensory system. It's observed in cortical areas, both in lower and higher cortical areas, that repeated stimulation gives rise to a decreasing response. This is just an example taken from recordings in monkey brain of initial responses and final responses after repeated stimulation, single neuron recordings, but you find it not only in single neuron recordings, in fMRI, in EEG, evoked potentials, all of this. It's called repetition suppression, and I haven't heard the word habituation referred to in any of that literature. And I'm completely puzzled as to why it is that neuroscientists of all people who you would expect are next door to cognitive psychologists would simply, they can't be unaware of the existence of the literature on habituation, why they basically don't even pay attention to it. Anyway, I mentioned this as, this is one of the minefields that I talked about. Habituation exists even in single mammalian cells. And it was Dan Koshland who did this beautiful series of experiments back in the 1990s with his students, again, showing habituation, showing that it had the characteristic features that Thompson and others had picked. He sets this very much in analogy to what's going on in neuroscience, and there have been exactly 0 references to Koshland's work in cell biology and signaling subsequently. Now, again, this is surprising because in the cell biology literature and the signaling literature, there's a very large literature on what is called receptor desensitization. And most of the time, when people do this, they, kind of give a step change of stimulus and look to see what happens downstream of the receptor. But every so often, you get people who do repetitive stimulation, and lo and behold, you get decreasing responses. And this is something similar. Let me not go into it. And again, nobody thinks about this as habituation. So habituation not only exists universally, it probably exists more universally than we're currently aware of, but we are not thinking about it as habituation. And then finally, we don't really understand how it works. And this is perhaps, for me, the most puzzling thing about all this, given that it goes back to the early 1900s that people have been observing habituation. You often hear the folk argument that, well, you're an organism, you're getting all these sensory stimuli, you decide that's not important, so you just decline to respond to it too much, right? I mean, that seems plausible, but that's really begging for some kind of normative theory that says an organism subject to all of these senses impinging upon it, and with limited resources, that's the optimal way to behave, to reduce your response in this successive fashion, and moreover, reduce it in such a way that these characteristic features of frequency dependence and intensity dependence are consequences of that optimal behavior. We have no such theory. Okay, so to me this is like, what? After 100 years? This is ridiculous. Perhaps if there was such a normative theory, we would be in a position to account for this remarkable universality that we see across organisms and scales. Quite apart from a normative theory, we don't even have a mechanism. Even for Koshland's 1990 experiments on habituation in PC12 cells, that's in mammalian cells. We know the receptors, we know the signaling pathways. Nobody's written down a mechanism and shown that it has the characteristics. So to our surprise, basically habituation is basically completely open. We don't understand it. It's everywhere. So if we're going to start thinking about learning, this is the place to start. All right, so let me just leave you with a few more kind of thoughts about why learning might be an interesting thing to do. I hope I've given you a sense that cognitive science offers us not only a sort of conceptual language, information theory, which is a very powerful theory. It is very quantitative. It gives us new measures for looking at things. But also, there's a sort of experimental paradigm that it gives us for looking at things differently. This question of the internal representations, the engram, I think to me is sort of a critical issue here. I really don't think that we will resolve the cognitive engram until this question of what the cellular engram is better understood. I don't think it's the full story, but I think it will be an essential part of the story. And what I haven't touched on here, and the thing that really sort of bothers me, it's all very well to talk about habituation. It's a very sort of universal feature of single stimulus learning. But if we are to treat learning seriously, then I think we have to set it. An organism, I would say that an organism is only going to learn what is evolutionarily significant to it. And what is significant to it has to be understood in an ecological context. Right? I mean, it's ecology that it's going to determine what it's interesting for it to learn. I learn mathematics. That's because I happen to be in a culture in which mathematics is actually valued. If I grew up somewhere else, it probably wouldn't be mathematics. I think the same thing happens to individual cells, which are organisms in their own right. They learn what is ecologically relevant. And therefore, if we are to take learning really seriously and get beyond sort of habituation as a sort of universal phenomenon, then I think we have to understand the ecological context much more deeply than we do now. And I think that ecological context is true not just of the organism, but of the cell within the multicellular organism. There is an ecological context in the developing organism. Okay, and let me finish by just thanking my partner in crime here, Sam Gershman, and also some neuroscience and cognitive science colleagues who formed a sort of informal discussion group for these kinds of crazy ideas, who've been very helpful in doing the thing that I find most difficult here, which is the transdisciplinary character of this whole thing. Because I'm a mathematician, I work in systems biology, and there's neuroscience, there's ethology, there's psychology, there's cognitive science. There's nobody who's an expert in all of these fields, and they all have very, very strong barriers against foreigners entering. And that's where the intellectual minefields are. So without some help, you kind of get torn to shreds. So I really thank the people who have helped me do all this disciplinary trespassing, and they're not responsible for the mines that I've set off. And a whole bunch of really fantastic students who have been naive, in the very best sense of the word, on taking on something which their elders and betters wouldn't touch with a barge pole. Thank you very much for your attention.
We have 9 minutes for questions. Ellen, and then...
Well, you could let James go first.
Sure.
Go ahead, Ellen.
Well, the trouble is I have way too much to say, so let me go first.
Okay, so I also had 2 questions, I'm afraid. So the first question, going back to your equation of developmental biology, the second term in that equation, the internal state, yeah. You seemed a bit reticent of saying that was learning within your framework, but it seems to me a good example, isn't it? It's learning, the cell is learning its position in the embryo from external cues. That's being processed through a GRN, which is a constant part, and then
using that after the cue has gone away. Yeah. So I absolutely I agree that it meets those criteria that I set out for learning.
So why were you reticent about calling it learning?
Well, I was reticent in the sense that developmental biologists don't usually use that word in thinking about what goes on inside the embryo. So the question is it useful to do that? And so I was thinking about this a little bit during your talk, James, and it seems to me that where it might be useful is that I think our current machinery, the machinery of gene regulatory networks, the machinery of dynamical systems, has been extremely good at giving us a formalization of the internal state of the cell, but not of the external environment in which the cell operates, which is for the most part wrapped up into parameters, right? So it's on the sort of, it's the boundary conditions that are set. And I think if we want to use learning in a more productive way, we have to formalize that environmental, which is also, of course, being produced by cells, as you pointed out. So then there's a sort of larger sort of dynamical system in the background, which is all the cells that are contributing to the construction of that. But one might not want to do the whole hog in one go. So I think the question that might be interesting there is, can we quantify the information that's present in the environment? And how much of that is actually being transferred into the cell? And I think that would be, for me, the starting point for trying to say something productive with the learning.
Can I ask my second question, which are unrelated? So the habituation, so that there seems to be some relationship between habituation and adaptation. Mm-hmm. Both, you could imagine it both in, phenomenologically, but also mechanistically. Mm-hmm. So has that
I think the word adaptation gets used in a lot of different ways, but in the homeostatic sense, it usually means returning the organism to the state it was in prior to the perturbation. Right, so there's some change.
That would be sort of a perfect adaptation.
Perfect adaptation would be a good example. Whereas habituation is somewhat different in saying that you ignore or successively ignore the stimulus that you're receiving. I think they both involve learning, and in the case of perfect adaptation, we can actually see what the internal model is. Your controller variable is a memory of the past history. So there is a form of learning that takes place, and the control theorists kind of understand that quite well.
And from a molecular mechanism point of view, that can be implemented by the induction of negative feedback regulators.
It can indeed, and in the case of, for instance, E. coli chemotaxis, which is a very well understood example of that integral controller, we actually have a very good hypothesis as to what the memory is. It's receptor methylation in that case. So that's an example where the engram is actually very close. And it's kind of, one could imagine that something similar might be at work in the context of, say, receptor desensitization or something like that. But again, that's not.
Okay, so this is more a discussion with you, but I think it's a useful discussion. First, I completely agree with you that this learning is occurring. It's just that the vocabulary used to describe it is different in different fields. An enormous amount is known about learning at the single cell level in T cell populations.
Oh, really? Okay.
Massive amount. It's just not called that because it's fine-grained so much that there are separate names for each of the different components of it. And it fulfills all your criteria perfectly. One of the beautiful things about T cells is that unlike the cells in most tissues, they are free to move.
Mm-hmm.
They sample multiple different environments.
Yeah.
And so you're actually looking at the cell's response, not only to a variety of environments, but just temporally separate re-encounters with the initial environment and seeing how the response changes.
Okay.
But there's an enormous amount known both about what changes in the individual T cell, what signals are needed to trigger different changes in the individual T cell, and then the conditionality of their activation by particular environmental cues to manifest either the desensitized state or the hypersensitized state. So I think that you would really enjoy getting into that literature because I think you'll find buckets of specifics about exactly the phenomena you're talking about.
That's wonderful. I would really love to get some more information about that.
Yeah, it's not the work that my own lab does the most, but it is incredibly rich in the T cell field.
So the word learning is, would an immunologist say that the T cell was learning?
They call it priming for memory, and so it's called priming, and there are forms of priming that either lead to memory or effector function, or something in between that's effector memory. Mm-hmm. And there's a series, it's well known that this is due to engagement not only of the receptor for antigen, but exactly which combination of cell surface receptors get engaged by environmental cues at the same time. So it's associative learning, but with a long-term, time-delayed decay, so that you then actually activate the negative feedbacks that James was just talking about as a function of the dwell time, which then gives you the upregulation of the inhibitory receptors, and those are actually involved then in making a cell susceptible to being turned off. But there are things that make it turned off, there are things that make it turn on more easily, that change the activatability of the T cell receptor.
And
so it's perfect for what you're
all this is
about.
taking place at the individual cell
At
level.
the single
It's not
cell
a
level,
process.
absolutely. And each individual cell is different. If you have one cell with the right T cell receptor to respond here, it doesn't matter what the other cells in the tissue are doing, it will basically follow this program in a certain way. So the other thing is, I don't think that you should worry about one representation of the engram. This comes back to what I was saying about Eric's view of the circuit elements themselves being functional units rather than components. Mm-hmm. In this case, you have different circuit elements than you would have, say, in a dictyostelium, discoideum, chemotaxide to cyclic AMP, mm-hmm. where you have similar things but totally different molecules. I think that it would be the circuit architecture. So I think there's a tremendous amount of support for your model, and happy hunting, and I'll be happy to show you my best ways to some happy hunting.
I would love to know. Thank you.
Sure.
That'd be fantastic.
The gentleman over there, if you have something short here.
Yeah. So I think you have mentioned that habituation is a universal phenomena. Is the mechanism of habituation is also same in different forms of life? No?
No, because it takes place in such different contexts. I mean, even at the organism level, if you think about the difference between vertebrate and invertebrate brains, there's a huge difference. And invertebrate brains are all different anyway. So even at that level, it's unlikely that the mechanisms will be the same. In the neuronal context, if you're thinking about the different forms of what the neuroscientists would call repetition suppression, again, it's occurring in different neuronal subsystems. And then you have habituation taking place in an individual cell. So I think the mechanisms are very different. It comes back to what Alan was saying. And I would say, this is a sort of comment on some of the things that were said earlier in the day. I think that a computer scientist would not find any of that surprising. Because in computer science, there's a very well, it's very well understood that you have this separation between function, algorithm, and implementation. And we all know that because, you know, you run the same web page on your iPhone and your iPad and your computer, and the architecture and the hardware is completely different. It's the same function. It's conceivably very different algorithms under the hood because they're different operating systems, they're different browsers. So I think if you grow up in a computer science world, this idea that, you know, the same function is implemented by completely different hardware is not a surprise. It's just normal. I think what's more surprising in biology is that that is embedded in the continuity of evolution, whereas there isn't a continuity of evolution in the technological setting. Right? So anyway, but yes, I think the mechanisms are very different.
Thank you very much.
Yeah, I find it fascinating and learning on the level of the cell. And that's when I was still a biologist, I learned that learning or engram has also to do with circuits, not Eric circuits, but circuits in the brain,
yeah.
And they happen between cells.
Yeah.
And so that cannot happen in one cell.
Well,
yes.
it cannot just happen in one cell. I think the neuroscientists, the current perspective has moved away from the purely synaptic view to the idea that memories are connected with ensembles of neurons that are sort of activated during memory processes. That doesn't mean that the synaptic view has gone away, it's just being added to with this higher level view of neuronal ensembles, but I think there's also a lower level set of things that are happening. There's a lower level set of things is what's happening in individual neurons as well. So I think if we want to tell the full story, there are several layers to it. There's a nuclear level, there's a synaptic level, there's a neuronal ensemble level, and we have to put all of those together to get it right. But there is a single cell level as well.
Okay. And only one semantic thing. At the beginning, you contrasted organisms with machines and pointed to learning. However, now we have machines that learn. And so that was maybe the 19th century view of machine, but in the end you said exactly something different. You know?
Yeah, so our perspective of the machine has changed most dramatically with the invention of the computer. But I think even the computer as a metaphor has changed from the computer of Turing's day to the computer that can implement deep learning and can drive your car, which we have now. Or beat us at Go, or teach us how to play Go. So that's a moving target. But I would say that we say machines that learn. I think that's not quite right. Machines that we program to learn. The cell learns without that. Yeah.
Just what I was thinking when you talk about development is exactly that the cells learn what they already know. It's like they're programmed to do morphogenesis. So the program is already there.
Like virtual learning.
Yeah, it's like they're programmed to what they call learn, but I don't know if it's really, like, I'm thinking of learning of something new.
But there is something new, because I would say, again, it comes back to my comment that learning has two components to it. There is the mechanism of learning, which, as you say, is programmed. I would say in the development, it is programmed. But then there's the content of learning. It is what is learned. And that can't be programmed, because every embryo is different. an individual cell is going to not know what its neighbors are until it finds out what its neighbors are.
It depends which kind of embryo.
Okay. Yeah. No, for sure. But even those kinds of embryos might conceivably have the odd accident. Right? I mean, they're, you know.
No, they define their neighbors very well, so there we go. Yeah, I'm sorry. I'm
No, but there's always this spectrum, right? So I would say, if you really ask the question, well, what does an individual cell in an embryo know? It has to know new things as its lineage is determined, right? In terms of who its neighbors are, what tissue it's going to become part of.
That's this signal, its program to respond exactly to this signal and make, you know?
Yeah. Well, okay, but I would say it's plausible that what it's programmed to do is to learn what to do in the context it finds itself in. But to me, speaking as an engineer, how would you design a cell that makes an organism? I mean, that's a really complicated thing to do, because the last thing you would want to do is to program it in the sense that you're saying, because it will never know for sure what situation it will encounter when it's doing it. It will always have to deal with the problem of stochasticity, always deal with the problem that while it's doing the right thing, its neighbors might have been too slow, or might have had an accident, or run into some problem, or died. And so it can't tell. So, it's like sending something off to the moon. We can't tell what it would. That's simple. That's a simple problem. We know what's on the moon, right? The developing embryo is like some other universe. So I would say, as an engineer, a much more robust way to do that is not to program it in the sense that you're saying, but to give it the ability to learn from its neighbors to do the right thing.
Unfortunately, we are way out of time. Thank you so much for the discussion. We can continue later during the dinner break. So we can move to the next session.